Hey everybody, it's Mr. Rogers, and I want to do a quick lesson on nucleic acids. And so we're going to go more into this later on in the class, so I just want to give a basic outline. So if it seems I'm skipping around, it's because we're going to come back to it and it's not a big deal. And so nucleic acids, their main job is up here. They store and transmit hereditary information, and they'll actually send it to the next generation. And they're your instructions. They're kind of like a cookbook. And we want to make sure we protect this cookbook because that cookbook cooks you. And if your cookbook is a little off and you're not cooking the right stuff, that could be a really bad thing. We could have very serious consequences to that. So we have to make sure that the DNA is structurally sound, we keep it safe, and that it can replicate and do things at the same time. Like, it's pretty crazy. You have the same DNA in all your trillions of cells, and we have to be able to replicate it all those times. And it's a pretty big process. And so the actual purpose of DNA is it's a code that actually ends up making proteins. And so the code that's written in that cookbook is making the proteins. Like I said, proteins basically make up everything else in your body besides the energy aspect of lipids and carbohydrates. And so this genetic information is going to code the proteins that make you. And there's two really famous, there's three, but really we're going to talk about two today. And that's DNA and RNA. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. And they have similarities. They have differences. RNA is older. DNA is newer. DNA is more stable. It's what we use, and we still use RNA though. And we use RNA as almost a messenger. DNA is something we keep safe, and then we read our DNA and we use RNA to send the signal to somebody else. And that kind of looks like this. You can kind of see we have DNA, we make RNA from it, and we send that RNA out because we don't want to send our DNA out into the crazy world. What if it gets damaged? And so we just send out this RNA little strip, and then we make a protein off of that code. And so you did that in 10th grade. That's nothing new. We're going to talk more about it this school year. So no worries there. And so nucleic acids are sometimes called polynucleotides. I'm going to be honest with you, outside of textbook, I've never heard anyone call nucleic acid a polynucleotide, but I at least have to say it to you. And then what you will have heard of before, and what we need to know, is the monomer for nucleic acids is nucleotides. And these nucleotides, there's 5 letters that you know of. There's actually more, but in this class we only have to worry about 5. And each nucleotide is made up of a nitrogenous base, a pentose sugar, which is the ribose, and a phosphate group. Okay, we learned about phosphate groups and functional groups. We kind of talked about ribose a little bit. You saw it when we talked about monomer sugars and whatnot. And then nitrogenous base is an amino group with some couple other things attached to it. Okay, so here, this is kind of what it looks like. We have the nitrogenous base up here. We have the sugar right here, phosphate group. And when we're connecting it, it's a constant connection. And this nitrogenous base is in the middle here. Okay. And this is one side of the DNA, or this could be just the RNA. And this is going to be in the middle, or the part that we're actually reading. This is the part that changes. There's different kinds of nitrogenous bases. The sugar and the phosphate group act as a backbone to connect the backbone going up. So this is kind of, think about this as kind of like carbon backbone, but it's not, obviously, but kind of like that. And these are like functional groups come off. Obviously not exactly, but similar. So these are the famous nitrogenous bases here that we're going to use. Sorry about my laundry going off there. We have cytosine, thymine, uracil, adenine, and guanine. You know these probably more as C, T, A, G, and then U is an RNA. T is only in DNA. Okay. I really don't hit this very hard, but I guess it's good to at least see. But the pyrimidines, that's cytosine, thymine, and uracil. They're single 6-celled rings. You see them over here. They're single 6-cell rings right here. And then purines, they're 5, and they're made up of 2 here. We have a 5 coming off of the 6. Okay. And I don't really talk about that too much. I really just kind of want you guys eventually to know that, A goes with T, C goes with G. And then when it's our RNA, uracil goes with A, U with A. The more important part is in DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. And the other thing that I really need you guys to get right now is for our knowledge, I guess we could do this one. So DNA is a double helix. That's a shape that you guys are pretty familiar with. DNA runs anti-parallel, which means that one side runs in one direction and the other side goes in the other direction. Think about a road. A road runs in anti-parallel. We have one traffic going one way and the other traffic going the other way. That's anti-parallel. So, for example, we have DNA always goes 5-3. We're not going to worry about that right now. That's later this course. But it runs from here, going this way. And the other side goes 5-3, runs this way, going this way. Okay? And so they run anti-parallel of each other. Let's see what else we can do. That's about it. I mean, the big thing, the other day right now, we just need to know about nucleic acids is they exist. They pass on information. They're made up of nucleotides. There's 5 nucleotides, and there's RNA and DNA. And we're going to talk a little bit about ATP as well tomorrow. But for now, these are the guys we know for nucleic acids. And it's pretty straightforward, pretty simple. If you need any help, let me know. And I'll talk to you guys soon.